Dear students, very good morning to all of you. Welcome in the world of science. Today, we will study about the fundamental unit of life, that is cell. Every body, every organism is made up of cell. Cells are the basic structure of life. It's a structural as well as functional unit of life. So, the branch of biology that is related to the study of cell is called cytology. Cells are very small structure organs, so we can't see them with the naked eyes. That's why we need advanced microscope to study cell. It was first observed by Robert Hooke, or simply the cell wall of dead cork cells, enclosing the vacant space left by dead cells. So all the credit goes to Robert Hooke and his discovery of cell in book entitled Micrographia. Okay, now what a living organism made up of? Every living organism made up of cell, just like a house made with numbers of bricks. A body is just like a house and cells are just like a brick. So we come on this discovery of cell. It was started from primitive microscope. Robert Hooke, a British scientist, observed a small compartment-like structure that was observed in the year 1665, become one of the most important milestones of the history of biology. You can say that the Robert Hooke left the foundation of cytology. Cell is a Latin word that means little room. It describes the structural unit of living organism is used till this day in biology. Robert Hooke was the first to prove that living thing consists of separate units. Okay. Now discovery related to cells. There are so many discoveries related to cells, just like Robert Hooke, Robert Brown, J.K. Perkin, J.T. Huxley, Rudolf Virchow, and Max Knoll and E. Ruska. There are so many theories these all scientists have propounded. You can go through this chapter. Okay, now what is the basic tool we need to study about the cell? And that is advanced microscope. Because we can't see cells by naked eyes, hence microscopes are essential for all cytological studies. Before studies, we have to follow some procedures like staining, mounting, slide preparation, fractional, etc. etc. These are the techniques that are used to study about cell. So now here we study about compound microscope. We need not to study about simple microscope because it's not useful here. So compound microscope having two main parts. One is mechanical parts and another is optical parts. Mechanical parts consist 9 portions, that is base or foot, first part on which a horseshoe shaped heavy basal part. Second one is arm or handle. It's a curve like structure to hold the microscope. Third is stage. It's a rectangular metallic plate attached to the arm, a hole at the center which allow the reflection of light rays to pass across. A specimen is kept on this stage. Fourth is coarse adjustment. It's adjustment below in low point or the draw tube move quickly on the coarse part. It is in the form of a small screw for gross focusing. Now the fifth one is fine adjustment. It's used to view the object under high power. We use fine adjustments. It's also a small screw-like structure. Stage clip. There's a pair of stage clips present on either side of a hole on the stage. These clips are used to hold the slide tightly. Nose piece, seventh part. It's a disc-like structure attached to the lower end of the body tube. It has three holes for objective lens of different magnification. There are body tube. Upper end of the arm is attached with the body tube that can be moved up and down. Its most important part of the microscope, the scale is the eyepiece at its upper. It is revolved nose piece fitted with three objective lenses and its lower end. The last one is diaphragm or condenser. The diaphragm is present just below the stage. It's used to collect and focus lights of the specimen. It controls the amount of light entering the microscope. Now the second part is optical part. Optical part consists only two main points. The first is lenses and second is mirror. There are two magnifying lenses. We use two magnifying lenses in compound microscope. That is why we call it as a compound microscope. If one lens is called simple microscope, more than one lens, that's called compound microscope. So the eyepiece lens may be 10x or 15x type, while 10x, 40x or 45x lenses are used to objective. The actual power of magnification of a compound microscope is to product of a compound of the eyepiece and the objective lens. Now second is mirror. There is a mirror mounted on the clamp attached to the lower end of the arm. It's a plane mirror mounted on the one side and a concave mirror on the other side. These mirrors are used to reflect the light rays. Simple phenomena. We can't use the compound microscope into a dim light or a closed room without light. Light is compulsory thing to use compound microscope. Now what is magnification? It's the power of an optical instrument to enlarge an object. कोई भी optical instrument object को कितना बड़ा करके दिखा सकता है, that is called magnification. The best quality compound microscope can provide us magnification of only 1000 times. Therefore, several components of the cells cannot be observed under compound microscope. To observe the ultra structure of the cell or the cell organ as electron microscope are used. Compound microscope, no doubt, simple microscope से बहुत ज़्यादा पावरफुल है, बट सेल्स की सारी ऑर्गेनेल्स को कंपाउंड माइक्रोस्कोप नहीं दिखा सकता. सो दैट अब हम इलेक्ट्रॉन माइक्रोस्कोप यूज करते हैं फॉर फर्दर डिटेल स्टडी. सो इट्स वेरी मच पावरफुल देन कंपाउंड माइक्रोस्कोप. ओके, now in next video we will study the further portion. Thank you.